Tyrosine. L-tyrosine or tyrosine symbol tyre or Y or 4-hydroxyphenylalanine is one of the 20 standard amino acids that are used by cells to synthesize proteins. It is a non-essential amino acid with a polar side group. The word tyrosine is from the Greek tyrós, meaning cheese, as it was first discovered in 1846 by German chemist Justus von Liebig in the protein casein from cheese. It is called tyrosyl when referred to as a functional group or side chain. While tyrosine is generally classified as a hydrophobic amino acid, it is more hydrophilic than phenylalanine. It is encoded by the codons UAC and UAU in messenger RNA. Functions. Aside from being a proteinogenic amino acid, tyrosine has a special role by virtue of the phenol functionality. It occurs in proteins that are part of signal transduction processes and functions as a receiver of phosphate groups that are transferred by way of protein kinases. Phosphorylation of the hydroxyl group can change the activity of the target protein or may form part of a signaling cascade via SH2 domain binding. A tyrosine residue also plays an important role in photosynthesis. In chloroplast's photosystem Roman II, it acts as an electron donor in the reduction of oxidized chlorophyll. In this process, it loses the hydrogen atom of its phenolic OH group. This radical is subsequently reduced in the photosystem Roman II by the 4-core manganese clusters. Dietary requirements and sources. The Dietary Reference Intake Recommended Dietary Allowance RDA for phenylalanine and tyrosine is 42 mg per kilogram of body weight. For a 70 kg person, this is 2.31 g phenylalanine plus tyrosine. Tyrosine, which can also be synthesized in the body from phenylalanine, is found in many high-protein food products such as chicken, turkey, fish, milk, yogurt, cottage cheese, peanuts, almonds, pumpkin seeds, sesame seeds, soy products, and lima beans, but also in avocados and bananas. For example, the white of an egg has about 250 mg per egg, while lean beef/lamb/pork/salmon/chicken/turkey contains about 1 g per 3 ounces 85 g portion. Biosynthesis. In plants and most microorganisms, tyre is produced via prophenate, an intermediate on the shikimate pathway. Prophenate is oxidatively decarboxylated with retention of the hydroxyl group to give p-hydroxyphenylpyruvate, which is transaminated using glutamate as the nitrogen source to give tyrosine and a ketoglutarate. Mammals synthesize tyrosine from the essential amino acid phenylalanine Phe, which is derived from food. The conversion of Phe to tyre is catalyzed by the enzyme phenylalanine hydroxylase, a monooxygenase. This enzyme catalyzes the reaction causing the addition of a hydroxyl group to the end of the 6-carbon aromatic ring of phenylalanine, such that it becomes tyrosine. Metabolism. Phosphorylation and sulfation. Some of the tyrosine residues can be tagged at the hydroxyl group with a phosphate group phosphorylated by protein kinases. In its phosphorylated form, tyrosine is called phosphotyrosine. Tyrosine phosphorylation is considered to be one of the key steps in signal transduction and regulation of enzymatic activity. Phosphotyrosine can be detected through specific antibodies. Tyrosine residues may also be modified by the addition of a sulfate group, a process known as tyrosine sulfation. Tyrosine sulfation is catalyzed by tyrosyl protein sulfotransferase TPST. Like the phosphotyrosine antibodies mentioned above, antibodies have recently been described that specifically detect sulfotyrosine. Precursor to neurotransmitters and hormones. In dopaminergic cells in the brain, tyrosine is converted to L-dopa by the enzyme tyrosine hydroxylase TH. TH is the rate-limiting enzyme involved in the synthesis of the neurotransmitter dopamine. Dopamine can then be converted into other catecholamines, such as norepinephrine noradrenaline and epinephrine adrenaline. The thyroid hormones triiodothyronine T3 and thyroxine T4 in the colloid of the thyroid are also derived from tyrosine. Precursor to alkaloids. The latex of Papaver somniferum, the opium poppy, has been shown to convert tyrosine into the alkaloid morphine, and the biosynthetic pathway has been established from tyrosine to morphine by using carbon-14 radio-labeled tyrosine to trace the in vivo synthetic. Precursor to naturalphenols. Tyrosine ammonia lyase TAL is an enzyme in the naturalphenols biosynthesis pathway. It transforms L-Tyrosine into p-coumaric acid. Precursor to pigments. Tyrosine is also the precursor to the pigment melanin. Role in coenzyme Q10 synthesis. Tyrosine or its precursor phenylalanine is needed to synthesize the benzoquinone structure, which forms part of coenzyme Q10. Degradation. The decomposition of L-tyrosine syn-para-hydroxyphenylalanine begins with an acetylglutarate-dependent transamination through the tyrosine transaminase to para-hydroxyphenylpyruvate. The positional description para, abbreviated P, mean that the hydroxyl group and side chain on the phenyl ring are across from each other. See the illustration below. The next oxidation step catalyzes by p-hydroxyphenylpyruvate dioxygenase and splitting off C-otwo homogentisate 25-dihydroxyphenyl-1 acetate. In order to split the aromatic ring of homogentisate, a further dioxygenase, homogentisate 12-dioxygenase, is required. Thereby, through the incorporation of a further otwo molecule, malolacetocetate is created. Fumarylacetocetate is created by malolacetocetate cis-trans isomerase through rotation of the carboxyl group created from the hydroxyl group via oxidation. This cis-trans isomerase contains glutathione as a coenzyme. Fumarylacetocetate is finally split by the enzyme fumarylacetocetate hydrolase through the addition of a water molecule. Thereby fumarate, also a metabolite of the citric acid cycle, and acetocetate 3-ketabutyrate are liberated. Acetocetate is a ketone body which is activated with 6-phenyl-CoA, and thereafter it can be converted into acetyl-CoA, which in turn can be oxidized by the citric acid cycle or be used for fatty acid synthesis. Fluoric acid is also a urinary metabolite of tyrosine in rats. Ortho and metatyrosine. 3 structural isomers of L-tyrosine are known. In addition to the common amino acid L-tyrosine, which is the para-isomer para-tyr, p-tyr or 4-hydroxyphenylalanine, there are 2 additional regioisomers, namely metatyrosine, also known as 3-hydroxyphenylalanine, L-m-tyrosine, and m-tyr, and orthotyrosine O-tyr or 2-hydroxyphenylalanine. The m-tyr, O-tyr isomers, which are rare, arise through non-enzymatic free radical hydroxylation of phenylalanine under conditions of oxidative stress. M-tyrosine and analogues, rare in nature but available synthetically, have shown application in Parkinson's disease, Alzheimer's disease, and arthritis. Medical use. Tyrosine is a precursor to neurotransmitters and increases plasma neurotransmitter levels, particularly dopamine and norepinephrine, but has little, if any, effect on mood in normal subjects. However, a number of studies have found tyrosine to be useful during conditions of stress, cold, fatigue in mice, prolonged work and sleep deprivation, with reductions in stress hormone levels, reductions in stress-induced weight loss seen in animal trials, and improvements in cognitive and physical performance seen in human trials. Tyrosine does not seem to have any significant effect on cognitive or physical performance in normal circumstances, but does help sustain working memory better during multitasking. Industrial synthesis. L-tyrosine and its derivatives L-dopa, melanin, phenylpropanoids, and others are used in pharmaceuticals. Dietary. 2 methods were formerly used to manufacture L-tyrosine. The first involves the extraction of the desired amino acid from protein hydrolysates using a chemical approach. The second utilizes enzymatic synthesis from phenolics, pyruvate, and ammonia through the use of tyrosine phenyl lyase. Advances in genetic engineering and the advent of industrial fermentation have shifted the synthesis of L-Tyrosine to the use of engineered strains of E. coli.